All right, so we are on Chapter 8, Section 2, and we're going to dive into our very first classification of receptor proteins, and those are G protein coupled receptors. So we're going to call them GPCRs now, right? G protein coupled receptors. So just so that we're all on the same page, there are going to be, I'm so sorry, lots of abbreviations like this. You just have to start practicing them. So these are super important to our bodies because they're involved in sensory perception, namely vision, taste, and smell. And they have an evolutionarily conserved structure, and that is 7 transmembrane alpha helices. And they have those 7 transmembrane alpha helices such that they're oriented where the amino terminus, right here, is on the extracellular space side, and the carboxy terminus is in the cytosol. So there's our carboxy terminus right there. And it's really interesting that most of these are glycoproteins, right? And they're glycoproteins because they have an extra carbohydrate moiety right here. So that's this looks like sugars, right, that are attached. And it helps their molecular recognition properties, right? And so when you look at these receptors, what you'll see is they'll start at the N terminus in the extracellular space. And then, so here they go, they start here. And then you go into these alpha helices. And a lot of times they'll be called serpent or serpentine receptors because what happens is they start in the extracellular space, they go down, right? There's a little loop and they come up, and here's the second helice, right? There's a loop between 2 and 3. 3 goes all the way along the membrane, comes up to 4. 4 will have a little loop to 5. 5 goes down. Here's our little loop to 6. 6 to 7, and then you get the C-terminal tail. So you see how it goes kind of up and down and up and down. So they call them serpentine. Sometimes depends on who it is. Let's see. I want to change this to single page. All right, much better. Okay, so the first G protein coupled receptor to be analyzed with X-ray crystallography was rhodopsin. And it was actually from squid, rhodopsin from squid, even though we all have rhodopsin. But it's a retinal molecule. And what's really cool about this particular G protein coupled receptor is that it doesn't react to a ligand in particular. It reacts to light. So it has a little recognition molecule kind of in the inner core of those helices. And so that when it is exposed to light will actually induce a conformational change in the receptor. And so the light signal is transduced to the cytosolic signaling proteins because of a change in the structure of the G protein receptor. So it's really cool, but it's not the most common. The most common is where you have a ligand that binds on the extracellular side, so out here, which then induces a conformational change down here in the cytosolic side. So what we're going to do first is we're going to watch an animation of how this G protein coupled receptor works. And this goes into lots of detail. And so then after we watch the animation, we'll talk about all the detail and how it all works. So let me see if I can switch over and do that. Okay, I want to make this big.
Our perception of vision, smell, and taste are all mediated in part by G protein coupled receptors, or GPCRs. GPCRs share an evolutionarily conserved structure consisting of a central core made up of 7 transmembrane alpha helices. In the cytosol, GPCRs interact with a membrane-bound protein complex called a heterotrimeric G protein, which consists of G alpha, G beta, and G gamma subunits. The G alpha subunit is a GTPase and can hydrolyze GTP to GDP. When the GPCR is not bound to the ligand, it is bound to the inactive GDP-bound G alpha beta gamma complex. In most GPCRs, an extracellular signal induces a conformational change in the GPCR protein. The conformational changes in the activated GPCR stimulate the exchange of GDP for GTP in the G-alpha subunit. GTP-bound G-alpha dissociates from the heterotrimeric complex. Both G-alpha and G-beta-gamma can then initiate distinct downstream signaling pathways. In some cells, different GPCR-mediated signals are integrated in response to different extracellular stimuli. Liver cells, for example, must respond to the hormones glucagon and epinephrine in order to regulate glucose levels in the bloodstream. Both glucagon and epinephrine activate a specific G-alpha called GS-alpha after binding to the glucagon receptor or the beta-2 adrenergic receptor. After exchanging GDP for GTP, GS-alpha undergoes a conformational change in a specific region of the protein called the switch II helix, so that it loses affinity for G-beta-gamma and interacts with an enzyme called adenylate cyclase. This interaction stimulates adenylate cyclase, resulting in the production of the second messenger molecule, cyclic AMP. At low levels of cyclic AMP, protein kinase A is bound to a regulatory subunit within an inactive tetrameric complex. As cyclic AMP levels rise, cyclic AMP molecules bind to the regulatory subunits, inducing a conformational change that releases the active protein kinase A monomers. Protein kinase A activation triggers several downstream pathways that all lead to glucose export into the bloodstream. One of the substrates for protein kinase A is the cytoplasmic tail of the beta-2 adrenergic receptor. The phosphorylation of the GPCR by protein kinase A and other regulatory kinases provides a docking site for beta-arrestin, a protein that results in downregulation of GPCR signaling and endocytosis of the receptor. The over 600 GPCRs in our body recognize a wide variety of signals, such as light, compounds we can taste and smell, and hormones, to enable our cells to perceive and react to environmental cues.
Okay, so I'm telling you that when we finish this lecture, I suggest you go back and rewatch that once we've kind of talked about all the different parts that they covered in detail. So it's a good thing to go back to. All right, so we're going to talk about a lot of different stuff in this section, and there are a lot of different abbreviations that are used. And so it's important for you to know what the abbreviations are, and I'm going to try to say them in their long-term and abbreviated form as much as possible, but I'm going to end up using some abbreviations. So this you can go back to, and you can see, oh, what she said pKa, what was pKa? pKa was protein kinase A, right? And it'll give you a little definition on its function. So this is kind of a nice thing to be able to go back to. So the first one that we're going to talk about in here is we're going to first start with the beta androgenic receptor. So BAR. It's not really B, it's really beta. And so this is a G protein receptor, okay? So when you have a ligand, and in this case, our ligand is epinephrine or, oh my gosh. The other word for epinephrine is adrenaline. A-I-N, adrenaline. I don't know how to spell it. I'm a terrible speller, but this is why they call it the androgenic receptor, because another word for epinephrine is adrenaline, but your book kind of sticks with epinephrine. So just kind of know that that's the same thing. So the beta-2 androgenic receptor, we call this beta AR or bar, right? So when epinephrine binds to this G protein coupled receptor, right, it's going to lead to a conformational change in the molecule. The ligand, the epinephrine, binds on the extracellular side. So here's the extracellular side. And it's going to induce that conformational change on the intracellular side. So if you look here, the blue is no ligand, the green is our ligand bound. And so you can see there's lots of conformational changes that occur over here in the intracellular side. And when this conformational change happens, now it's going to affect interactions of the receptor with intracellular signaling proteins. And so it's those new interactions that are going to lead to changes in activity of all kinds of different things that we're going to talk about. And so knowing how this works, we can actually design drugs to regulate G protein receptor activity, right? And we can design drugs in two different ways. We can design receptor agonists. An agonist is a receptor that will, it's a signaling molecule that mimics the natural ligand response, right? So if you have a drug that binds to your beta-2 androgenic receptor, it's going to result in the same thing happening just as if epinephrine were binding. So it acts just like, or mimics, the natural ligand. An antagonist is going to bind to the receptor with high affinity and prevent binding of the normal ligand. And when it binds, the cool thing is that it's not going to induce a structural change. And so you're not going to get signal transduction. So they can be used to basically make the receptor more active. That's this one. Or they can be made to inhibit activity of the receptor. So either way, which is kind of interesting. So if you look, here are some examples of natural, these are our body's natural ligands. And then here are two different kinds of pharmaceuticals, and these are agonists and these are antagonists, right? And so the isoperotenol, I'm not even going to try. It's albuterol. It's albuterol. This one is albuterol. So if you know someone who has asthma, right, this is used by asthmatics to relax the smooth muscle in your lungs and to open airway passages. And it works very rapidly, works very, very well. But it also happens when you're in fight or flight, right? And you make epinephrine. If you make epinephrine, the whole point is to get away. Something is terrifying you. You are in extreme high stress conditions. There's there's a bear that just entered the classroom, right? You are freaked out. And so you want to be able to get as much oxygen into your body as possible. And so you're going to relax the smooth muscle in your lungs. You're going to open your airway passages. You're going to get more air and you're going to be able to run faster and get away from the bear, right? So it's the same thing. The other two over here that are antagonists, these two are going to actually inhibit a signaling pathway. And so these inhibit the signaling pathway that control vasoconstriction. So these inhibit vasoconstriction. So this is inhibiting the opposite process, but in essence is going to do the same thing, right? So these two can actually use to treat, you're going to treat high blood pressure, right? If you inhibit vasoconstriction, those blood vessels are going to relax. They call them alpha blockers, right? And they can be used to treat high blood pressure. Same thing on this agonist. This agonist can be used to treat high blood pressure, but for the opposite reason, right? Because it mimics the normal things in the body that dilate the blood vessels. So opposite mechanism, same result. Okay. So we have a lot of different kinds of signaling with our GPCR receptors in metabolism, right? And so there are two different hormones that we're going to talk about real quick. And you should kind of know about these hormones a little bit. So glucagon is I'm hungry. This is what your body makes when you have low blood sugar, right? And the whole point is to get your body to put some glucose into the bloodstream, right? Now, epinephrine, like we said before, is that fight or flight, fight or flight, right? Either you fight the bear or you run from the bear, right? And so this hormone is released from the adrenal gland. but only in times of really, really acute stress. And so it'll increase your heart rate, it increases your metabolism, all that kind of, you need more energy, right? And so you're gonna get that energy through breakdown of glycogen from the liver, from lipid breakdown, anywhere where you can get energy because you are absolutely terrified and you need some sugar in your bloodstream so your muscles can work, right? So each of these different hormones kind of wants the same effect. We want to put glucose in the bloodstream. And so they do it in different ways. So they each bind to a type of GPCR receptor, right? Glucagon binds to the glucagon receptor. Epinephrine can bind to two. Epinephrine binds to the beta-2 androgenic receptor and the alpha-1 androgenic receptor. And so depending on which GPCR is activated, you're going to have different downstream effects. And so you kind of skip to this last column here, right? These are different alpha subunits of the G protein. So remember, right, you have that receptor, and the receptor was bound to the trimeric, the heterotrimeric G proteins that were alpha, beta, and gamma. Well, they don't just make one alpha. We have different kinds of alpha subunits. You have S subunits, which are stimulatory. You have Q, you have all kinds of different ones, right? And depending on which one is stimulated, then you're going to activate a certain kind of enzyme. When that enzyme is activated, it's going to induce release of a particular secondary messenger. And so if you're glucagon, right, you're going to activate the G alpha stimulating G protein. And so you're going to result in adenylate cyclase activation. Well, guess what? If you're epinephrine, even though you're going to go through a different receptor, you're still going to activate a G stimulatory subunit. And so you're still going to activate adenylate cyclase to make cyclic AMP. And so both pathways are different, but they're going to get to the same end result. And so we call these shared pathways. So these two are shared pathways, right? Now, epinephrine, we said, can bind to two different G protein coupled receptors. So it binds to these two, and each of these two also activates a different kind of G protein subunit. And so it could be the GS alpha or the GQ alpha, right? And so they're going to activate different enzymes. One's the adenylate cyclase and one's phospholipase C, or PLC. Right? So if you activate phospholipase C is going to cleave that membrane phospholipid into DAG and IP3, right? That was from the last unit. So both signaling processes are going to have the same result, but they're going to go through different pathways. So you put epinephrine in, and no matter which way you go, you're going to end up turning your glycogen to glucose. So the very end of the pathway is the same. Everything from start to finish is going to be different, but the very end of the pathway is the same. And so what we call those parallel pathways. Okay, so they are parallel pathways. So these two here are parallel, and this is just kind of an animation of that. And then these two here are the shared pathways. Okay, and so you can see in the end, everything's going for the same goal. We're going to get net glucose export into the bloodstream, but depending on which signaling pathway you go through, you're going to get there a different way. So just like I was saying that you have many types of proteins, right? You're going to start with kind of a main scheme where you have receptor activation with kind of a ligand, right? Kind of a ligand, really a ligand. So that ligand, right, is going to be bound to your receptor here. And it's going to induce that conformational change in the GPCR in that cytosolic region. So over here, you're going to have kind of a conformational change. What happens when that conformational change occurs is you have a receptor-mediated stimulation where we replace GDP by GTP in the alpha subunit. So right here in the alpha subunit, we start off with GDP, we're going to end up with GTP. When that conversion occurs, basically the trimer disassociates. And so now we have an alpha subunit, and then we have a beta gamma subunit, okay? And then from there, many things can be different. We just call it downstream signaling, and we're going to talk about that difference. So it's important that you understand the steps that are going to occur for GPCR activation, which should have been here, and I'll go back to that next slide, right? But you have ligand-induced conformational changes in your receptor. Then you have receptor-mediated stimulation of the guanine nucleotide exchange. So you go from GDP to GTP, and this is in G alpha. And then you have regulation of all of our downstream things by this G alpha that's bound to GTP and our free G beta and gamma complex. And then eventually we're going to have to talk about how that signal is terminated, because we haven't really discussed that yet. But all right, so there are many different types of each of these subunits of the G proteins. So the G alpha is a GTPase. The beta and gamma are not. Only the alpha is a GTPase. The GTPase has the ability to hydrolyze GTP into GDP, right? And so for each type of G protein, of G alpha protein, right, you have distinct downstream pathways. We talked about the GS alpha. This activates adenylate cyclase. We're going to talk about that today quite a bit. But you could also inhibit adenylate cyclase if it's a different G protein subunit, right? It's a different variation. You can stimulate phospholipases, right? All kinds of different things. So I just kind of want you to know that they have different variations of these subunits and they can do different things. Okay, so when we talk about why these G protein coupled receptors are important, right? They're important for our senses. Why are they important for our senses? Well, it's all about neuronal transmission, right? It's going to require our G protein coupled receptor signaling, right? And so if you look at them, the whole reason that they're different is because they're regulating levels of different secondary messenger molecules. So here in A, right, we have light being absorbed by a retinol group, right? And that group in rhodopsin activates our GT alpha subunit. This stimulates a phosphodiesterase, right? Remember we said it has different downstream effects. And you have cyclic GMP hydrolysis. So that causes here our cyclic GMP hydrolysis to GMP, right? And that actually causes an ion channel closure. And when you have that ion channel closure, now you're going to have neuronal signaling. So that's how we know we've detected light, right? We have to convert some physical thing into a signal to the brain. And so that's how we do it for vision. For scent, for olfactory receptor, you're going to have some kind of odorant, right? A particle. That's what this pink thing is. And it's going to bind to our olfactory receptor. It's going to stimulate the GS alpha G protein. That's going to stimulate a adenylate cyclase. An adenylate cyclase, right? The adenylate cyclase is going to convert ATP to cyclic AMP, right? The cyclic AMP is going to cause ion channel, in this case, ion channel opening. When that ion channel opens, we're going to induce neuronal signaling. Oh, look at that. We're going to end up at the same place, right? Same thing when we have taste and olfactory. Not olfactory, um, gustatory, right? Gustatory binding. So if you have binding of something sweet, right, and it binds to the taste receptors on your tongue cells, it's going to activate the GQ alpha G protein. That's going to stimulate phospholipase C. When phospholipase C is activated, we're going to cleave our PIP2 into DAG and IP3. When that happens, these two are going to cause an ion channel closure, which then induces a neuronal signal. So all of this is required for us to be able to have our senses, which are really pretty nice, right? Okay, so let's get into a little bit more detail. So remember in the last section, we discussed signal amplification, right? We had our receptor that was activated, and it led to activation, just like here, whoops, just like right here with our adenylate cyclase, right? And we converted ATP to cyclic AMP. And then in this case, we kind of went into more detail and we said the cyclic AMP activates the pKa, right? When we have active pKa, our pKa is going to phosphorylate our phosphorylase kinase. So remember this is where we're talking about all the fold increase in the signal. And so when you have that phosphorylase kinase is going to phosphorylate our glycogen phosphorylase. And the glycogen phosphorylase converts glycogen to glucose 1-phosphate, right? So I'm trying to get you to remember what pKa does, right? So it's a downstream signaling protein, and it's active when we have high levels of cyclic AMP, right? Because that's what activates pKa. So when that happens, we have actually 3 distinct metabolic responses, right? Why would we want pKa to be active right now, right? What we want is we want to put glucose into the bloodstream, and we're actually going to do it in 3 different ways, which is absolutely awesome that the body is designed this way. We're going to phosphorylate and we're going to inhibit glycogen synthesis. If we can stop glycogen synthesis, that's really great, because glycogen synthesis is taking glucose and turning it into glycogen for storage. We want to stop making glycogen. Stop making glycogen. That's really important, right? If you want to put glucose in the bloodstream, you got to stop making glycogen. It's also going to phosphorylate and activate enzymes that are involved in glycogen degradation to produce glucose, right? So this is great. Whoa. We're going to start converting glycogen to glucose. This is what we want, right? Woohoo! But at the same time, you're also going to activate enzymes through phosphorylation that are involved in gluconeogenesis. Oh, what are we doing? We're going to make glucose. So you have to have, I kind of like to think about it as a seesaw, right? When one side goes up, the other side's going to go down. And so you have to inhibit the conversion of glucose to glycogen at the same time as you need to promote the degradation of glycogen to glucose. So when one goes up, the other must go down. It's a really great mechanism for really tight control on activities inside of our bodies, right? So let's kind of look at a diagram of how that works, right? So we have blinding of glucagon. This is our I'm hungry, I haven't had anything to eat in a while, to our glucagon receptor. And it actually could also be, in this case, epinephrine binding to a beta-2 androgenic receptor. Remember, these are shared pathways. So either way would stimulate this pathway. And so when we have that, we're gonna have our GS alpha that's gonna convert GDP to GTP. We're gonna activate our adenylate cyclase. When we activate our adenylate cyclase, we convert ATP to cyclic AMP. Now we have cyclic AMP. Cyclic AMP is going to activate our protein kinase A. Our protein kinase A is going to go through 3 different things. It's going to turn off glycogen synthesis, it's going to turn on glycogen degradation, and it's going to turn on glucose synthesis. All in all 3 things are going to result in a net export of glucose into the bloodstream. And that's what we want. That's exactly what we want. Okay. All right. So what if we are epinephrine, but we're not going to bind to the beta 2 receptor. We want to bind to alpha 1. What happens? Okay. Well, we're going to end up at the same result, but we're taking a different path, right? So in this case, when we convert our GQ alpha, right, we're going to convert that to the GDB to the GTP version. We're going to activate PLC, right? PLC is what, phospholipase C is what hydrolyzes PIP2. When we hydrolyze that membrane phospholipid PIP2, it's going to generate our secondary messengers, right? Our secondary messengers are DAG and IP3. So when we have DAG and IP3, what are they going to do? Well, the DAG is going to activate PKC, right? So that's what we have here, this little arrow here. DAG activation of PKC, so that's great. But also our IP3 is going to induce calcium release from the endoplasmic reticulum. So that's going to initiate glycogen degradation. But at the same time, it's going to also, you have that calcium that was released from the ER, that's going to further activate our PKC. So when we have that calcium, that's going to also activate PKC, and then together that is going to promote basically the off switch for glycogen synthesis, right? So end result, net glucose export, and that's what we want. So you just kind of have to go back over these kind of diagrams and see that you can follow along and that kind of thing. But this is really great, really, really great. But how do we terminate a signal? Once you've started this, how do you make it stop? Because you can think about it, if you can't make it stop, it's going to go out of control. So there are really 2 different ways. And one of the ways is the G protein cycle, and the other way is receptor desensitization. And so that's the next one we'll talk about. So our G protein cycle is really the sequential stimulations by 2 different things, GEFs and GAPs. Okay, what are they? GEFs are guanine nucleotide exchange factors, right? And these are the things that promote that conversion from GDP to GTP, right? They activate our signaling. And that's exactly what we saw in that animation, right? We saw that they were able to induce this conversion, right there, where we had GDP and now we have GTP. So now our signaling molecule, our G protein, is active and it is doing its thing. We got signaling, we're going to have lots of downstream things happen. That's great. Okay, how do we make it stop? Well, we have what are called GAPs. GAPs are GTPase activating proteins. GAPs are going to stimulate the GTP hydrolyzing activity of a G protein. So we're going to change from having GTP to having GDP, right? So GTP to GDP. So this is what is going to inhibit signal transduction, right? And in your book, right? So they said, okay, here's your GAP, here's your GTPase activating protein. We're also going to call it by another abbreviation because you just need another one. So we're going to call it RGS. What is RGS? RGS is regulator of G protein signaling. And they just call it this because it's a GTPase activating protein that specifically functions with, so it's specific, PECR, specific to G protein coupled receptors that have G proteins. So you should probably know what that abbreviation is. It's just so many abbreviations in this one. I'm so sorry. Okay, so the other way that you can do this is through desensitization. So after our alpha dissociates from beta and gamma, right? You can have this G protein coupled receptor kinase that phosphorylates the cytoplasmic domain of our G protein coupled receptor. And so when it becomes phosphorylated, basically it's going to mark it. This is marked for recycling. So and I'm going to show you a little animation, not an animation, a diagram, right? Okay, so here we have, let's start at number 1, right? We have a ligand activation of our G protein coupled signaling. And in this example, we're going to use the beta-2 androgenic receptor, right? And we have it coupled to our alpha beta gamma trimeric protein here. Okay, so what happens is you're going to induce that signaling, right? So you have GPR signaling. We're not drawing that in this diagram. But now, whoa, okay, sorry. But now we have this additional protein. And so we have this receptor kinase enzyme that's called the beta androgenic receptor kinase, or BARK. BARK is recruited to the membrane with the dimeric gamma, G beta gamma complex, right? So this is my G beta gamma complex. And so that's what it gets associated with. So this phosphorylates our GPCR. Okay, so they don't show it, which is kind of not nice, but it becomes phosphorylated, right? So now that this thing is phosphorylated, oh, they do show, okay. So now that it's phosphorylated, I guess I was just behind, right? Now that it becomes phosphorylated, this is a docking site, and it allows another protein to bind, and the other protein is called beta-arrestin. Beta-arrestin is going to bind right here to this phosphorylated terminal end of our receptor. Okay, well, so what's beta-arrestin going to do? Well, it's going to prevent the receptor from reassociating and making that G alpha beta gamma complex, right? So 2 things can happen. Either the beta-arrestin GPCR complex can be internalized and put into a vesicle, right? And then beta-arrestin can go and bind to another G protein complex and go again, right? Or once that thing is internalized, it can be broken down. It can be recycled. It can be degraded and the parts can be used, or we can take it, leave it in the vesicle, but then put it back into the membrane when we're ready. So it can kind of go 2 different ways. Either you can just store it inside a vesicle in the cell for a little while, and when you're ready, put it back out on the membrane, or you can break it down. And it just kind of depends on what your goal is. But the whole point of this is that we can control the duration and the strength. Control duration and strength of whatever extracellular signal we have. By having a termination event. And so that's super, super important so things don't spiral out of control. So I do suggest you go back and rewatch that animation at this point. All right, y'all have a great day.